Jenna, can you hear me?
Yep, I can hear you.
Good. Okay. Vodka. Oh, vodka. That stuff is rock good. Vodka, it's awful.
I've never really drank vodka, so I wouldn't know. But actually, it's funny. So we have this in Albania. We make this. Well, you probably have some stores like we call it raki, but it's
Raki. Yeah. That's Greek.
It's
made from prunes.
You can make it from any. So they make the wine and then they distill what's left over.
Oh, okay, yeah.
Really high alcohol content. We would use that. Or whatever it's called.
The two national drinks of Croatia. In fact, Eden has probably had this. Kruškova. It's a pear. Kruška in Croat is pear. Kruškova we have at Christmas. Delicious. And then Slivovitz, plum brandy. I don't like that. But Kruškova. Yeah. Kruškova.
Is that similar?
It's a pear. It's a pear brandy.
Yeah, I mean, they can make that with.
It's sweet. It's rather sweet.
Oh, this isn't sweet. Great. Cool. Yeah. Like you all had steak. My mom would do this stuff. What is yours called? I still can't stand the smell of it. Yeah. What's it called? What is it? It's called Glycolysis. RAKI. Here's my everyone's little like line. And I'll just put all those around that for a while.
It's time.
Oh no!
Thank you.
Feel better?
Yeah.
Good.
Thank you. Okay.
Yeah, do close that, okay?
reagents? Agents?
Reducing agents that are reduced, oxidizing reagents that are oxidized. Yeah,
Oxidizing agents?
Agents. agents, agents.
So I'm doing a abstract for ChemChem. Is the footnote number go after the period or before the period?
Goes after,
Oh, after. Okay.
after, yeah,
And then it would go before a comma.
after a comma.
It would go after a comma.
Yeah.
Okay, thank you.
Okay. All right. Back to glycolysis. So to review, we have glucose. It's phosphorylated. We use ATP to make glucose 6-phosphate. Because of the hydrolysis of ATP, this reaction is highly exergonic. The next step, we rearrange that to fructose 6-phosphate. That makes the phosphorylation of the number 6 carbon, or the number 1 carbon, excuse me, much more easily accomplished. Incidentally, this particular protein, phosphohexosaisomerase, can be secreted from the cell, and if it is, it's a neurotrophin. It drives nerve growth, the growth of neurons, which is kind of interesting. And then the next step is the most highly regulated step in glycolysis. You phosphorylate carbon number 1, make fructose 1,6-bisphosphate. You use phosphofructokinase 1, and that's a highly regulated enzyme. One of the allosteric activators is fructose 2,6-bisphosphate. That metabolite is made by PFK2, and there are lots of things that regulate PFK1. Then we take this molecule apart via a aldol condensation that's worked in reverse. And this is the enzyme aldolase. Aldolase will take fructose 1,6-bisphosphate and will degrade it, I guess you could say, to 2,3 phosphocarbon compounds, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate. The delta G for this reaction is not so hot, as you can see. However, the end products are removed quickly. Because they're removed quickly, that will push this reaction through. There are 2 types of aldolases. They're called very creatively type 1 and type 2. Type 1 aldolases do not require a divalent cation, and they use a Schiff base mechanism. Type 1 aldolases. Type 2 aldolases are found predominantly in microorganisms. They contain a zinc cation, metal ion. There's also a conserved arginine that will be involved in substrate binding. That enzyme is different than the type 1 aldolase we have in our bodies. So the type 1 aldolase uses a Schiff base mechanism. That's shown here. Let's make this a little bigger so it's easier to see. So you have a lysine residue that is absolutely conserved in these type 1 aldolases. The binding pocket will put the lysine very close to the carbonyl carbon. These lone electrons here, these extra pair of electrons, will be able to interact with that slightly positively charged carbonyl carbon. This keto oxygen will abstract this hydrogen here. There's an acceptor group on the other side of the substrate binding pocket. You also have 2 basic groups. Those will be important in just a bit. You then make this amine intermediate. This hydrogen will be abstracted by the basic group down here. That basic group's identity changes from enzyme to enzyme.
Hey, Dr. Baranovich, your slides aren't moving.
Slides aren't moving. Of course. Yes. Now is it moving?
No, they're not.
I don't know what to do then. Then do you have the slides in front of you?
Yeah, I can just put them in front of me.
Then just look at them because I don't know what to do. All right, so this, we'll take this hydrogen here, this basic group, that's going to liberate a water, and you're going to get a shift base out of this. Here's your shift base for which the intermediate is known. This basic group has the hydrogen from the lysine here, and now you have a bond to that carbon. Now, since you have a bond to that carbon, you're then going to have these electrons here rearrange. This hydrogen will be abstracted by this basic group here. Now, as we can see, this part here is going to take its electrons and leave. So because you've abstracted that hydrogen, you make an aldehyde, you break that bond here. That's your first product that's released, glyceraldehyde 3-phosphate. This compound here is now going to have this hydrogen. This is your enamine intermediate. That hydrogen, as you can see here, is going to be given to this oxygen here. Well, actually, it's going to be given to that bond there. That's going to create the alcohol here. This, then, you're going to have a water come in. The water will break this bond here and will provide you with a ketone here. And it's also going to provide the hydrogens that are missing for the lysine and for this acceptor group here. So you get from the water the oxygen here, hydrogen here, hydrogen here. That releases a second product. So this is a ping pong mechanism. The ping pong mechanism. You bind the first, you release first substrate, release first product, bind the second substrate, water, release the second product. Ping pong mechanism. You've now restored the enzyme to its former glory, if you will, and now it's ready to accept another substrate. That's the shift base mechanism that you see for the type 1 aldolases. There are also 3 types of type 1 aldolases in our body. They're called very creatively A, B, and C. A, B, and C. So A is expressed in muscle and red blood cells. Muscle and red blood cells. B is primarily expressed in liver, but also in kidney, and also the small intestine. If you have a mutation in the aldolase B gene, you have a fructose intolerance. Fructose intolerance. An inherited fructose intolerance. For B. Aldolase C is expressed in the brain, but also in smooth muscle, platelets, and other types of tissue cells around the body. Sorry?
The A, B, and C are all type 1.
They're all type 1. Mm-hmm. Then the final stage, the preparative phase, is the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. That is triose phosphate isomerase. Triose phosphate isomerase, regardless of where you see it, in the world of biology, has a set of beta sheets that form a ring with alpha helices on the outside. So it forms the so-called TIM barrel. A TIM barrel. This delta G is not very favorable either, but the payoff phase is going to grab that glyceraldehyde 3-phosphate and get rid of it lickety-split. Yeah, delta G/0, still positive, but you can push it through because the end product concentration will be very low. Now we come to the payoff phase. We are going to take glyceraldehyde 3-phosphate. We're going to convert it to the very high energy 1,3-bis phosphoglycerate. Bis phosphoglycerate. Now, how do you convert an aldehyde into a carboxylic acid? You oxidize it. So, if you're oxidizing it, somebody has to accept electrons, right? So you're going to get a reduced NAD from this reaction. This reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate dehydrogenase. This is an NADH utilizing enzyme. It has a cysteine residue at the active site. And you can derivatize that cysteine and kill the enzyme if you use certain compounds. So it oxidizes the aldehyde, and it will make a so-called acyl phosphate. Acyl phosphates are pretty high energy molecules. So you take inorganic phosphate, glyceraldehyde 3-phosphate, you're going to remove reducing equivalent here and here. You will make NADH, and you'll generate 1,3-bis phosphoglycerate. Very high energy compound. So you're going to form a thiohemiacetal. Thiohemiacetal, that's going to be the intermediate that you're going to use to complete this reaction. So here's the mechanism of glyceraldehyde 3-phosphate dehydrogenase, sometimes called G3PDH or GAPDH, either one. Mechanism's the same. So here is your active site. Now, in this enzyme, you have an NAD that's actually covalently bound to the molecule. You have an NAD that's covalently bound to the molecule. It's at the active site. This is unusual. You see it in some enzymes, not very often. Usually NAD is not covalently bound. This one is. Here's your cysteine. So glyceraldehyde 3-phosphate binds. Here's the aldehyde carbon. Here's the negatively charged sulfur. And at this site, the pKa of that cysteine is very low. So you get a sulfide. Now, sulfide is very reactive. Reacts with the carbon, and it forms the hemisulfide thiohemiacetal, right there. That's the thiohemiacetal. This reducing equivalent here will go to the NAD and make NADH. All right, so this is what's this? Thioester, high energy. Now you're gonna have soluble NAD that will come in and relieve the enzyme bound NAD of its reducing equivalent. So soluble NAD comes in with the phosphate. You get a NADH that comes out. The phosphate is going to attack here. So it's going to break the bond here. That's called phosphorylasis. Phosphorylasis. Or phosphorylasis, depending upon where you accent the syllable. Phosphorylasis will add the phosphate here. You're going to make an acyl phosphate. That breaks the bond here. Now you've regenerated the enzyme. Nanda? Phosphorylasis, yeah. You have phosphorylasis occurring from here to here, step 5. Step 5. And you can see it there, phosphorylasis. That's the mechanism of glyceraldehyde 3-phosphate dehydrogenase. Very, very interesting enzyme. So you form the thiohemiacetal. You then get oxidation of the thiohemiacetal. You make a thioester. You cleave it via nucleophilic attack by the inorganic phosphate. That results in phosphorylasis. That phosphorylasis releases the acyl phosphate, which is the glycerol 1,3-bis phosphate. Again, this is really high energy. Very, very high energy. And if you use the right reagent, like iodoacetate, it'll derivatize the cysteine and it'll kill the enzyme. So iodoacetate can be used and it will form an adduct with the cysteine and you'll kill the enzyme. That's one of the ways you can tell if you have a cysteine at the active site. Give it iodoacetate. If iodoacetate kills it, you know you have a cysteine there. What kind of inhibition is this? This is irreversible enzymatic inhibition. So you make 1,3-bis phosphoglycerate. You're now going to use that high energy molecule to make ATP. You're going to give the acyl phosphate to ADP to make ATP. And the enzyme that'll make ATP and 3-phosphoglycerate, this enzyme is called phosphoglycerate kinase. Phosphoglycerate kinase. So here's ADP. There's that high energy phosphate. You're going to take that and stick it here. That gives you 3-phosphoglycerate. So if you can draw 1,3-bisphosphoglycerate, just erase the phosphate and you have 3-phosphoglycerate. Notice this reaction is very, very exothermic. That's going to help pull those other not very favorable reactions forward. So this is ATP synthesis by substrate level phosphorylation. You make a high energy phosphate compound. You then take that high energy phosphate bond, high energy carbon phosphate bond, usually, and then just transfer it to ADP to make ATP. Substrate level phosphorylation. So 1,3-bisphosphoglycerate is such a compound. You donate the acyl phosphate to ADP to make ATP. The kinases, they're your enzymes that transfer that phosphate from ATP to something else or from something else to ADP to make ATP. That's why this is a phosphoglycerate kinase. Phosphoglycerate kinase. Very favorable. Very favorable. And because of its coupling to glyceraldehyde triphosphate dehydrogenase, this reaction is virtually irreversible. Third step, now you're going to take 3-phosphoglycerate and rearrange it. Oh, it's reversible, sorry. It's reversible because in gluconeogenesis, you're going to run this backwards. Sorry, don't even read my own slides anymore. Okay, now that phosphate, you're going to take it off and put it back on. You're going to put it on carbon number 2. So this is phosphoglycerate mutase. Phosphoglycerate mutase. That will catalyze this reaction. So this takes this phosphate, takes it off, sticks it here. And you use magnesium as a cofactor. So this enzyme has a very interesting mechanism. So it requires a cofactor that is 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate. And that cofactor is made just by phosphorylating 3-phosphoglycerate by virtue of an ATP-dependent kinase. So you make 2,3-bisphosphoglycerate, or BPG. And here's how this particular enzyme does its business. So you have a phosphohistidine. The phosphohistidine takes this off, sticks it here. Now you have 2,3-bisphosphoglycerate. Then you're going to take this phosphate off and stick it back here. So you're trading one for the other. So let's say you label this phosphate. That labeled phosphate ends up here. And then the phosphate that ends up back there ends up being unlabeled if you use an unlabeled substrate. Or if you use a labeled substrate, the phosphate that comes off is unlabeled and your label ends up being back in the enzyme. Very curious mechanism.
So what makes it attack the bottom phosphate? They look identical.
Yeah, they do. The positioning of it in the enzyme. Yeah. Yeah. Same one. Same one. Now you're going to take 2-phosphoglycerate and you're going to pull out of water. And you're going to make phosphoenol pyruvate by pulling out that water. And this is the enzyme enolase. Enolase. So you take these two here, that's water, take them off, generate a double bond, you make an enol group, so it's phosphoenol pyruvate. Phosphoenol pyruvate. And the very last step of glycolysis, you're going to take that very high energy phosphate, transfer it to ADP, make ATP. And this is the enzyme pyruvate kinase. Pyruvate kinase. And this, look at that. And that is going to pull the rest of glycolysis down into the sink, essentially. Because this delta G is so very negative. And because pyruvate tends to get used pretty quickly, that's just going to take the whole thing and pull it right through. Pyruvate can tautomerize. It will tautomerize between the keto and the enol form. The keto form is a predominant form at pH 7. That's the form that will therefore predominate inside cells. But the tautomerization is going to take essentially pyruvate. It's going to make the end product very, very stable. That further drives a reaction to completion. And I have a handout that gives you the whole thing on one sheet of paper, front and back. I have one that goes on paper like this, but it's really small. So I figured this was a little easier to read.
Sure. Do you want to like a graph and I could take one and pass that down?
Yeah, you can pass those down that way. And then I'll give 2 to Selma, and then give 4 to Do, 3 to Telly. All right. So, again, let me emphasize, how did I learn this? I learned it by drawing it out multiple times and making mistakes multiple times. And then fixing my mistakes and going back and drawing it again. Was it tedious? Yes. Was it time consuming? Yes. Did I want to quit? Yes. But I got it done and I learned it. If I learned it, you can learn it too. So start now. Start now. So it'll take you a while. And you have other pathways to learn too. Not just this one. Now, in some bacteria, there is an alternative route to glycolysis. It's only found in some bacteria. It's actually pretty widely distributed amongst these bacteria. But this is called the Entner-Duderoff pathway. Entner-Duderoff pathway. I spell that right. D-O-U-D-O-R. Yeah, spelled it right. Now, the payoff phase of the Entner-Duderoff pathway is identical to glycolysis. And the Entner-Duderoff pathway begins the same way that the Ebden-Meyerhoff-Parnas pathway begins. You phosphorylate glucose and you make glucose 6-phosphate. The change starts with the second step. You have a phosphogluconate dehydrogenase. Phosphogluconate dehydrogenase. That is going to then lead to a dehydration step. So you have a phosphogluconate dehydratase, phosphogluconate dehydratase. That leads you to this compound that has this formidable name of 2-keto-3-deoxy-6-phosphogluconate. 2-keto-3-deoxy-6-phosphogluconate, which is mercifully abbreviated KDPG. KDPG. I would. Then you have an unusual enzyme that's called KDPG aldolase. That will form glyceraldehyde 3-phosphate and pyruvate. Interestingly, the KDGP aldolase has a alpha-beta barrel structure as well, just like your other aldolase. So the glyceraldehyde 3-phosphate goes through the payoff stage, and you get 2 pyruvates, but ATP-wise, you're only going to make 1 net ATP, not 2. Just 1 ATP, not 2. Why do cells use this instead of the more common pathway? I have no idea. They do. And some of them grow pretty fast. So they just do. That is a variation of glycolysis. The last part is all the same. The differences are these 3 reactions right here. Well, none that I can see. All right, so you have the classic pathway of glycolysis. Then you have essentially this semi-phosphorylative sort of thing, and then this is just essentially another way to show you essentially how you skin that cat, essentially. Some of the cells that use the NAD pathway have a glyceraldehyde 3-phosphate that makes an NADPH, and NADPH is used for biosynthesis. So this is a pathway that gives you something more for biosynthesis than for something else. That might be one of the reasons why cells might use it. Beyond that, I really can't tell you. Now, as we said, all the ATP made during glycolysis is made via substrate level phosphorylation. And if you make all your ATP via substrate level phosphorylation, then you have a metabolism that is fermentative, and your cells undergo fermentation. So yeast at the bottom of the big vat that has the barley or the grapes or the whatever, those yeast are deep in the throes of a highly anaerobic situation. The only thing they know how to do is ferment. They will ferment and make ethanol. Do all fermenting organisms make ethanol? No. No, no, 1,000 times no. I was at a church service where someone said there were no yeast in the Garden of Eden because yeast, ethanol came after the fall. So there was no fermentation in Eden. I'm sitting there saying, all that leaf litter at the bottom of the tree there, and there's no fermentation going on? Excuse me, but I have a question. And I asked him afterwards, I said, just asking, are you aware that there are many types of fermentation and only a fraction of them make ethanol? He said, no, but all fermentation came after the fall. Right. So horses then and cows weren't able to eat before the fall. Or were they carnivores? No. All carnivory came after the fall. Well, how do they digest all that plant fiber? Because you know what does it in their gut? Microbes. You know how they do it? Fermentation. I'm confused. Oh, you're a smart aleck, aren't you? No, I'm just confused. So, and I ended the conversation there because it was clear he was getting pretty mad. So some arguments just aren't worth having because some people just aren't willing to listen. And when I was your age, I wanted to convince everybody and I got really mad. And then I got a little bit older and I discovered that some people come to conclusions for lots of different reasons. Facts are usually only a part of that. Sometimes it's a very small part of that. So just keep that in mind. Okay, let's now take a look at the fate of pyruvate. All right, Jenna, let's up. Gotta make sure that's there. Make sure that's ended. All right, try it again. Try it again. There we go. There we go. Yes, it can be taught. Okay, ladies, are the slides moving? Yes or no?
Yep, they're moving.
They're moving. Okay, now let's try this. Are they moving now? Yes or no?
No, I don't think so.
No?
Nope, they're not.
Yep. They're just not going to move. I don't know why. Okay. So if we were to take glycolysis at a glance, we take 2 glucose, we expend 2 ATP, we generate 4 ATP, 2 NADHs, and 2 pyruvates. That's what we would generate. And so we get 2 ATP per glyceraldehyde triphosphate. 2 plus 2 is 4. 4 minus 2 is 2. So our net ATP is 2. Net ATP is 2 per glucose. So organisms that ferment aren't going to grow very fast. That's one thing you should know right from the get-go. Now, you have a reduced NADH. You have two reduced NADHs. So if we just sum up the whole thing, those are the reactions. Glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphates go through glycolysis. You get 2 pyruvates, 2 reduced NADs, 2 hydrogen ions go into solution, 2 ATP plus 2 waters. That's essentially the reaction, summing it up at a glance. And if we simplify it into half reactions, we'd have this half reaction that has that delta G/0, and we'd have that one, whose delta G/0 would be 61. And so we get an overall delta G/0 of minus 85 kilojoules per mole. Very, very exergonic. We get our ATP through high energy phosphorylated intermediates. That means we make all our ATP via substrate level phosphorylation. These phosphorylated entities, at pH 7, they're charged. They're not going to leave the cell. They can't diffuse out of the cell. So essentially, once you phosphorylate things, they're stuck. They're not going to move. Now, let's talk about the problem that glycolysis gives us. So that's pyruvate. You now have reduced NAD. If you keep running glycolysis, all your NADH is going to end up as reduced NAD. But to run glycolysis, you need oxidized NAD. So how do you return this to this? If you are fermenting, you're usually going to take this, you're going to dump those electrons here on your carbon skeleton. So if you were to take, for example, these reducing equivalents, and you were to give it to pyruvate, you would generate and who's that? Lactic acid. Lactic acid. That is essentially a lactic acid fermentation right there. So that's one way to skin the metabolic cat. The other way is to take pyruvate and get rid of this. And if you do that, you get this. Who's this? Acetaldehyde. If you reduce this, you get who's that? Oh, guys, if you don't know what this is, ethanol. Thank you. Ethanol. I'm going to tell on you to Dr. Baldwin if you don't know that. All right, so you could see how you could potentially make lots of different compounds by essentially tweaking the sorts of things that you get from glycolysis. What you get from glycolysis is pyruvate, but there's plenty of ways to take pyruvate and do interesting things with it. And dump your electrons on it. You can make things like acetone. You can make butyrate. You can make propionate. You can make lots of different compounds via fermentation. This is the reason why saying ethanol equals fermentation is just not true. Ethanol fermentation is actually extremely varied, diverse, and extremely interesting.
Is that why you like fermented something wrong instead of getting alcohol, you get vinegar?
Yeah. And if you use the wrong microorganisms, yeah. Yeah. This leads me to a little story about when I was in graduate school. So we read a paper, and there's a, first of all, little introduction. There's a fungus that infects the central nervous system. It's called Cryptococcus neoformans. Very nasty fungal infection. You get it from pigeon dung. And this outfit in Boston said that they could detect Cryptococcus by looking for ethanol in this cerebrospinal fluid, because when the organism gets in your body, it grows like a yeast. So they said yeast make ethanol. So we looked at that paper and we said, I don't think so. So there are ways to grow Cryptococcus in a yeast in the laboratory. And when we did that, we then took the culture media and we put it through a gas chromatograph and we asked what came out. We discovered there was a little bit of ethanol, very little. There were lots of other things. There was so little ethanol, there was no way you could use it diagnostically. So we published a little follow-up communication to that paper, and we said, yeah, we grew the organism as a yeast, checked out what it was making while fermenting, and it made a little ethanol, but not enough to detect in cerebrospinal fluid, that's for sure. Sorry?
What were they detecting?
They were detecting ethanol in cerebrospinal fluid, and they're saying that that was a diagnostic test for Cryptococcus. We said balderdash. Even with a heavy infection, there was no way you're going to have enough ethanol within the CSF to detect that. So please don't make the ethanol equals fermentation equals ethanol mistake. People have made that mistake and they have paid for it. All right, so let's move on now to something a bit weightier, shall we say.
So they were detecting ethanol, but it wasn't necessarily from the fungus.
No, yeah, exactly. You can detect ethanol from other things in CSF. One thing they didn't do a healthy control. If they did a healthy control, there's a little bit of ethanol there actually. And in fact, it wasn't too far different from their Cryptococcus. So yeah, folks, do your controls. Do your controls. All right. So you want to reoxidize NADH. You don't do that, metabolism will grind to an inglorious halt. So let's look at ways to do that. So first, you just have lactic acid fermentation. You just dump it straight onto the pyruvate. The enzyme lactate dehydrogenase will catalyze that reaction. Lactate dehydrogenase is found everywhere. Very, very ubiquitous enzyme. And this is the basis of lactic acid bacteria. They perform a lactic acid fermentation, and in fact, Streptococcus mutans, which sticks to your teeth by means of an extrant capsule, it sits there and it makes lots of lactic acid, and it etches your teeth. That's how it does it, via this fermentation. And lactic acid bacteria aren't the only one that do this. Plenty of other bacteria do this. This is also responsible for the organisms that spoil milk. You ever taken that milk out of the fridge? You notice it has that really, really off smell. Then you taste it, that is lactic acid. That's lactic acid that's causing that off taste, that off smell in the milk. Yep. Yeah. Yep. Yep, pretty foul. Pretty foul. Now, guys, this is actually useful industrially. So how many of you like sourdough bread? What is that particular taste in sourdough bread? It's lactic acid. Because lactic acid fermenters help make sourdough bread. How many of you like yogurt? Yogurt is made by a lactic acid fermentation. That's why the pH is low. That's why its tendency to spoil is low. Cheeses, other types of stuff. So this is useful industrially too, as well. Not to mention your muscles when they get into this aerobic fermentation as well. Okay, let's pick this up on Monday. Have a great weekend. Pray for us. We have a quiz tomorrow. John 4, 5, and 6. Okay, folks. That's all for today. See you guys on Monday.